gets mutated. So I know we talked about during replication how faithful DNA polymerase is, but there are other ways that mutations can get introduced. So what is a mutation, right? So this is going to be a change in your genetic material. But this is going to be different than most things we've talked about so far, because now this is a heritable change in the sequence, okay? We are changing the sequence of our DNA. It's permanent, it's passed on. That's a mutation. So if it's a change in the genetic sequence, which mutations can be mutated? By and large, your answer should be DNA. To a lesser extent, it can also be RNA when we think about an RNA genome in viruses. And we know that viruses acquire lots and lots of mutations over time. We can talk about the broad consequences of mutations. So what effects do they have? Well, this is twofold. One, they can be beneficial because we know that mutations are the source of our genetic variability and that mutations allow evolution to occur. On the other hand, they can be detrimental because they lead to diseases and disorders, right? Now, we can put mutations in several categories. One is based on size. So we talked about chromosomal mutations at large a couple chapters ago. Now we're going to focus on these gene mutations. These can be as small as one base pair change, okay, versus an entire chromosome. These, because they're one base pair, usually affects one gene, and you can only detect it by sequencing that gene. All right. And now this lecture is going to go over the different types. We can also classify mutations by the type of cells that they occur in, so somatic versus germline. And you should be very familiar with the differences in these two by now. And so think about it. If it's a somatic mutation, how does it affect an organism? If it's a germline mutation, how does it affect an organism? And we will revisit this. So far, so good? So let's talk about these effects. So what happens when we have these mutations? How do they change the structure of a gene? How do they change the function of a gene? In general, we're going to talk about point mutations. So these are single base pair changes. These single base pair changes can occur in two types, a base substitution or a single insertion deletion. So insertion deletions are point mutations only when they involve the insertion or deletion of one base pair. So our base substitution is substituting one base for another. So essentially we have changed this C to a T. That's a single base pair change. That's a base substitution. There are two types, and it's based on what we're substituting. So remember that our nitrogenous bases can be classified as pyrimidines and purines. Pyrimidines are the single ring, purines are the double ring. So our pyrimidines are C, U, and T. Our purines are A and G. So if a base substitution is a transition, it's substituting a purine for a purine or a pyrimidine for a pyrimidine. Okay, so these are the more common of the two. The second one is a transversion. So now we're going from a purine to a pyrimidine or a pyrimidine to a purine. So A to C or T, G to C or T. So we are changing that category. So if I give you a sequence, you should be able to introduce a base substitution, right? So for example, if this T became a G, now we have a base substitution. Which flavor? Is it a transition or a transversion? Okay, so your answer should be this is a transversion because we went from a T to a G. So if it helps for you to make a DNA sequence, make a DNA sequence. Or you can also use a sentence. And why are my sentences arranged in threes? Why are my sentences consist of three-letter words? You should have an answer for that. So a base substitution would be changing one. So instead of saying the big cat had fun, we can say the big rat had fun. And that would be a substitution. So we changed one base pair. Now we're going to talk about insertions and deletions, also known as indels. These occur in two types. Are they frame shift or are they in frame? So think about the reading frame. So when we talk about the frame here, we are referring to the reading frame. So if it is a frame shift, it's a non-multiple 3. Why? Because our reading frame is in codons or every 3 nucleotides. Okay? So If it's an in frame, we're sticking in or taking out 3, 6, 9, and so on. And the effects we will see vary drastically in our protein sequence, okay? So if it's a frame shift, you insert 1, the entire reading frame is changed. If it's an in frame, you insert 3s, you don't change the entire frame shift. You just insert additional codons, which means inserting additional amino acids. Okay, so an example here, you can write this sequence down and add or subtract 1 base pair, add or subtract 3 base pairs, and see what happens. If you look at the sentence, if it's an in frame, you will put in a word. So instead of the big cat had fun, the big bad cat had fun. If it's a frame shift and you take 1 out, everything changes. This is no longer coherent. So our next flavor is these expanding nucleotide repeats. So we have talked about these before. And what happens is these trinucleotide repeats are inserted over and over again. Okay. So here we had a CAG, right? CAG. Now we're inserting CAG, 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 CAG. So which non-Mendelian inheritance pattern is associated with trinucleotide repeats? You should be able to come up with that answer like that. Because remember, we're building on what we learned, not learning, take an exam, forgetting. So give me an example of an expanding nucleotide repeat. You would just pick 3 base pairs in here and repeat them over and over again. And if it was in a sentence, the big cat had fun, 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 fun. Okay? So far, so good.